Everyone see the screen?
Yes.
Hey, everyone, thank you for coming to this month's monthly online seminar. This month we're talking about metabolic modeling. We're going to hear from Dr. König and Dr. Hatzimanikatis. So quick, first just some background about the seminar. So the motivation behind the seminar series is that there are a number of people working on biological modeling, but they're geographically distributed. And it's important for us to come together to share ideas and brainstorm solutions to things, but it's difficult to do that physically. So the goal of the seminar is to bring everyone together virtually. But as of course you guys know, for the past few months, it wouldn't really matter where everyone lives, because this is still, this is currently the best form of communication. Okay, so for the structure of the series, the, large-scale structure has been that each month we focus on a specific biological subprocess and how it gets modeled. And the rationale behind that is that for each biological subprocess on its own, there are some unique challenges and therefore some unique methodologies. So there's a lot of overlap between, say, modeling gene expression or modeling metabolism or cancer biology. But each one of these subprocesses, there are unique challenges to them, and therefore it's, I think, good to talk about their own methodology. And then we can think about how the different methodologies relate to each other. For each seminar, the structure is that we hear from two presenters, each one for about 20 minutes, and that's followed by a group discussion where people can ask questions to the presenters or just general questions about the topic to brainstorm with everyone else. Okay, so the goals of the group discussion are to identify common challenges, share personal solutions, brainstorm new solutions, and of course, to help create a collaborative community of modelers. The rules for the discussion are: one, err on the side of commenting. If you're not sure whether others will be interested in your comment, the overwhelming odds, I think, are that they will be. You should just go ahead and say it. Two, honest debate is always encouraged. Just be polite. Sometimes I wish we had more debate, but you have something, just go ahead and say it. And three, mute your microphone when you're not talking, though this rule has been followed better lately. Okay, so this month we're talking about metabolism. Okay, so just to give sort of an oversimplified view of metabolism. So the way I've always come to think of metabolic models is like one biological subprocess that takes place. So there's the dogma of DNA gets turned into RNA, and then RNA into protein, and then protein expression relates to the metabolism, and then in turn the metabolism affects the RNA and protein concentrations. Okay, this is an oversimplified view, and I think in a sense the way we ourselves have structured our seminar series kind of implies this way of thinking, because we had one series on RNA modeling, another one on protein signaling modeling, and this month we're talking about metabolic modeling. But I think it's important to keep in mind that there's sort of a difference in scale here. So we can think of metabolism as like one process that needs to be modeled, but it's really many sub-processes. So as an example, like this is a metabolic network of a number of different processes like carbohydrate metabolism, nucleotide metabolism. I think in many ways each one of these, like smaller sub-processes, can rival the scale and complexity of something like translation or transcription. So it's important to keep in mind that metabolism is hugely complex, and it itself comprises a lot of smaller but still enormous sub-processes. And I think another important difference to keep in mind, or I shouldn't say necessarily difference, but okay, you could argue on this, but I think that when you think about metabolic modeling, arguably it requires a more integrated approach compared to other biological sub-processes, like a protein signaling network. And then that's because if you're focused on the pool of metabolites, any other metabolic sub-process will affect the metabolic pool, and that will in turn affect many other metabolic sub-processes which draw on the same metabolites. So I think in general, this would encourage a more integrated approach, because if you want to look at how I guess, different metabolic subprocesses relate to metabolites, you need to keep in mind how other subprocesses are using those same metabolites. Right. Okay, and yeah. Okay, so so there's, I think, a unique challenge for metabolic modeling, which is collecting data for these models. Well, this is always a challenge, but I think it's an especially hard challenge for metabolic modeling. And this is because, so the two types of data you would likely need to create a metabolic model is metabolite abundance and reaction kinetics. And both of these data types are less prevalent compared to things like proteomic or transcriptomic data. So you can find much more data on protein and RNA abundances than you can on metabolite abundances and reaction kinetics. And this makes constructing these models very difficult because it's just hard to get the data necessary. Two, in addition, metabolic models can be enormous. And this creates, a difficult problem just because the scale of the models can be a lot bigger. There are many more parameters that you would need data to parameterize. Okay, so let's go over an overview of current methods of metabolic modeling. The two most common ones are kinetic modeling, or it's basically a system of differential equations. So compared to the other method, this is relatively more precise, but it requires both knowledge of the metabolic network and knowledge of the rates of each reaction. And these models can be dynamic, like they can occur over time because you have the rates of each reaction. Okay, then at the other end, you can have flux balance analysis, and compared to kinetic modeling, this is less precise. But the advantage is that it only requires knowledge of the reaction network. You don't need to input the rate of each reaction. And the way this essentially works is it infers the reaction rates based on a given objective function. One of the most common ones is growth. So somehow you'll mathematically define growth, and then it will set the rates of each reaction to optimize maximum growth. And this can be further refined by adding constraints to the reaction rates. Yeah, and in generally, these models are static. So as opposed to kinetic modeling, which you look at distinct time points, flux balance analysis, I guess in general, really look at like an entire cell cycle, or it won't have distinct points in time. So with that overview, I would like to introduce our first speaker, Dr. König.
Thanks a lot. I just have to share my screen. Okay, can you see that? Yeah. So, welcome everybody. Today I will talk mainly about reproducible modeling of liver function. What we're mainly interested in my group is to find out what the liver function of a given patient is. And this is a really important question because you need this information in diagnostics of liver diseases, in monitoring of these diseases, and also in the monitoring of progression or the effect of possible interventions. And very important question for liver function is basically the functional capacity, which means like how much is the liver able to fulfill a certain function. Perhaps like one step back, why the liver and metabolism? The liver is basically the metabolic hub of the body, and it's responsible for most of the xenobiotic reactions, meaning detoxification of foreign substances, but has also a central function in glucose homeostasis and fat metabolism and protein and amino acid turnover and so on. So many people consider this as a metabolic hub. Especially interesting, it is the liver has capacity for very different metabolic pathways, like the core pathways we saw on this introductory map, but also many of these detoxification pathways. So how do we want to measure measure and to model liver function. So the tool that we mainly using is so-called dynamical liver function test. What is done here is basically that to a patient, a certain test substance is given, and then over time in the plasma or other body fluids, the decline of the substance over time can be measured, and this can be used as a proxy for liver function. Of course, there are also test substances where then some metabolic product is measured, then a curve would just a second metabolized product would appear, and this could also be used as a proxy for the liver function. This is a very interesting approach because you can use very different test substances. Like classical examples for that are caffeine, metacetine, which is used in something called the Limax test, but also galactose, codeine, midazolam, and so on. So like there are many different test substances, and most of these are metabolized by different pathways. So there is not really one single liver function, but there's like different pathways which have different functional capacity, and these can be tested with different test substances. This is a very interesting approach because it gives basically dynamical information on actual liver function in vivo. But the approach has some challenges. The first one is that one has very high intra-individual variability in such tests, meaning if you give the same amount of the test substance, it's very different how these curves look in different subjects. There are many reasons for that, but it's just like it creates basically the problem that what does the test curve mean? Like what is basically the actual liver function and what is like this intra-individual variability? The second thing is that the whole metabolization is highly dose dependent. Okay, so how do we approach this? And the main idea is that we build something like, we build multi-scale models which allow basically to describe the whole body physiology. And in this whole body physiology, we can put different organs. Mainly we put in the liver, but also like mainly the kidneys, the two main organs which are responsible for the detoxification of these test substances. This is basically I would say like a template model, which we then can initialize with different metabolic models for our given test substances. So it's basically, like, I would say, a multiplex approach. You have the different test substances, which are going via different pathways, and you can just basically reuse your whole body physiology in your computer model, and you just put in different metabolic pathways for your test substances of interest. Of course, like in the long run, you could also combine these via like intermediate metabolites and so on. But for now, these are basically like a separate approach for the separate test substances. And if you have done this whole body models with liver metabolism in the liver and also kidney metabolism within the kidney, then you can individualize and stratify such models by accounting for things like age, gender, body weight, or lifestyle factors like smoking and so on. So how do these models look most of the time? It's I would say like compartment-based ODE models where the different body compartments are connected via the blood flow, and then in certain submodels we can basically via hierarchical model composition put different metabolic networks, and here we put it in the liver. What are the data sets which we are using for the parameterization and for the validation of such models? Basically plasma concentration time curves, but also information about appearing metabolites in the urine or in saliva and so on. Basically any body fluid which can be measured, but normally routinely like plasma measurements, serum measurements, or urine measurements are taken in patients. So we get this time curves, like for instance here it's for caffeine and one of the products paraxanthine, and then we can use this this input for our models, or like often what is reported also parameters calculated on these curves. So in the literature, not these curves are reported, but basically they calculate like an elimination rate or a clearance or area under curve from these curves, and only this secondary parameters calculated from the time courses are reported. And these are basically the two main data we use for the modeling. So I would say 5 main problems go into this model building. One is like standardized data, the reproducible models, software for modeling, compute infrastructure, and in the end, we want to bring all this together to get actual applications. So first thing we had to build was basically a large database of this pharmacokinetic information, basically finding a way to store this time course data and also like storing the calculated parameters from that. And for that, we did basically a large literature research, digitized a lot of information from the literature, put this in the standard data format, and integrated this with all the patient information which was reported in the papers. We did a lot of semantic annotations so that we automatically can bring together this information with our computational models. We did this, I mean, this is an older slide, but around 500 studies right now, and have, I would say, around 20,000 to 50,000 pharmacokinetics, but also we have like actually like a few thousand time courses right now, which we can use for model building. Yeah, we have information studies groups, individuals, interventions, pharmacokinetic parameters, time courses, and references. Importantly, we have all the data for the stratification of this data sets, and also then for the stratification basically of the computational models. We did some meta-analysis of the data. For instance, here in this simple plot, we can then see basically every data point is a single study from a different group. Like the small points are individual data points. And here the whole thing is then stratified by some information. Here's basically the smoking and oral contraceptive status of the which was reported. And we can see the smokers have basically higher clearance than the oral contraceptive users. Yeah, we can do this for more information. Okay. Quick overview how this looks. Then basically we have then this large database. We can there like this just search for the substances we want. You can see like for different studies, different things are reported. And then basically digitize a lot of data, all the patient information, like for the individuals. Basically, if individuals are reported, we know like their oral contraceptive use, age, and so on and so on. And then also like here, like time course data. Okay, this was not so interesting, but there are lots of these data which we can directly then integrate and get basically this time course data. Okay, this is our database for the metabolic modeling. Next step is basically like some reproducible model building, which is really important. We have all our model infrastructure based on SBML. We're using basically SBML core and the SBML comp to build this hierarchical models, combining organs with the whole body physiology. Have a lot of unit validation, unit checking, unit conversion. It's like very important. All our data sets and all our models are completely unit validated, and annotated, which then allows the automatic unit conversion and mapping of data sets on our computational models. We use a bit of SBML distrib for uncertainties. And by having SBML, we can use a wide range of tools for simulations, parameter fitting, and so on. As simulator, we use basically Roadrunner. I would say a bit of Tellurium, but basically SBML sim, which is like some utility functions, I would call, or it's more than utility functions right now, but something around Roadrunner, which allows like doing the simulations. If we have the model in SBML, we can also do like automatic things like, or we can have tools for automatic model building, for automatic model checking, and automatic model visualization, which we all build on top of SBML. The next building block we need, like for this metabolic model, is basically cloud infrastructure. A lot of these simulations are very intensive, so we have to do like a lot of parameter scans, a lot of parameter fitting, a lot of Monte Carlo approaches, and all this is very intensive. So all our tools are running basically in the cloud, and so our workflows have been Dockerized and just run on Kubernetes infrastructure. So we can, as we have in-house clusters and different clusters, and if we need more compute infrastructure, we just buy some AWS. And we can just put our workflow like via Kubernetes in the different infrastructures, which is very nice. Yeah, exactly. We built an in-house cluster, but there's basically also running Kubernetes, but it has some specialized functions for certain problems. Okay. I don't have much time, so I will just run a bit through one of the use cases, but it just shows, okay, how this is generally done. Like this is basically a metabolic model for the caffeine, which was then stratified and used for individualized predictions of risk. like I showed the model structure before, like on the whole body, this is very simple. And then you build not too complex metabolic networks, most of the time, just a handful of reactions in the liver and in the kidney. And by that, you are able to model the different substances. Then you automatically map the data from the database on our models and create simulation experiments from that. Basically, from the database, we read, okay, what was the dose in application for? In this case, for instance, 4.94 milligrams per kilogram body weight were given orally. You can automatically create simulation experiments for that, do like simulations with parameter sensitivity analysis, and just automatically create the plots from our model. So basically, our model is adapted on the body weights, which are reported in the study. The dosing is automatically reported, and the data sets are mapped based on the annotations on the model and automatically unit converted, because every study reports different units, and by having units everywhere, we can bring this together. And then we do this for many things, basically like many curves, and then we're doing some model predictions. Here's, for instance, the prediction now of the mean model for the different stratified caffeine pharmacokinetics curves. And we see like that a mean model in black is not very good in predicting this large variability in the different subgroups. The subgroups are here smokers or contraceptive users or combinations of these. And we can just see the mean model is not very good. And then we can basically go and adapt our metabolic models by going in the literature, figuring out what is going on in the subgroups. And basically the protein amounts are induced or inhibited, and so the distributions of protein amounts is shifting in these different subgroups. If we account for that in the model, basically just saying, okay, the main enzymes are distributions which are shifted by the lifestyle factors, we then are much better able to predict this different subsets. And that we can of course also do then for the pharmacokinetic parameters, and can predict then the dose dependency of this clearance. So this is basically the applications. We can then have a lot of validation data, because we only build our models on subsets of the time course data, but we can use all the pharmacokinetics data which is in there, and for many different studies we can just use for validation, and then pretty good. Same we can do for individualized predictions. Okay, this is not so important, but if we have individual data and have the individual lifestyle factors, we can put for the individuals the changes in the parameters in the model, and then basically the main info is here, like the more information we get for the patient and we have like the model trained for it, the better the predictions get. Okay, I still need 5 minutes if I can. I wanted to, in the end, like present like some how we in the end like publish basically our models. And we developed something which is called EXecutable SImulation MOdels, called EXSIMO. And the main idea, I would say, is like the main motivation is from this quotation that basically publications are only advertisement, and the actual scholarship is a complete set of instructions and data which generated the figures. And so I tried to implement this and basically just create all, like record all the instructions and data which generated the figures. And this basically was in the end like something like called an EXSIMO, which combines version control with the automatic model building, with annotations, units, automatic testing, automatic reporting in an iterative cycle of creating new model versions, and then can automatically basically create all the simulation outputs or simulation experiments which were run in the studies. We use a lot of version control, create a ton of tests to ensure correct behavior, and automatize basically the model building, analysis, and reporting process. And in the beginning, we built this basically for, in the end, to publish some reproducible thing. But now it's basically how we build our models, because we're getting direct feedback what is wrong with the model and can iteratively improve our model all the time, which is very nice. So yeah, like I said, like a lot of tests, for instance, annotations, do they exist? Are they correct? Do the units exist? Are they correct? Can the model be built? Basically, the whole model building process is part of the pipeline. It's not we have a model and we run something with it, but the whole model building pipeline is part of this reproducibility. Model checking model can be simulated, checking data files exist, the data fields can be processed, the unit conversions are working, all simulations can be run, and reports can be generated. And then we have very interesting things that we have all this automatic unit conversions, and basically we can just have to say our plot should be in this units, and all the data and all the models are converted in this unit automatically. And this can be basically run on any computer. It's just Dockerized. You have one command, and it's basically reproducible on any system. Yeah, a lot of people help with that. I would say mainly in the data curation, and I hope it was not too fast, and I didn't take too long. If you have any questions, please ask. Thanks a lot.
Okay. Thank you, Dr. König. Okay, so I noticed there are a lot of really interesting questions that are in the comments. So we're going to wait until after both presenters have presented, and then I guess if either the one who asked the question can just go ahead and ask, or I'll read off the questions. But yeah, we'll do it once both of them are done. So so I would like to introduce Dr. Hatzimanikatis.
You're muted.
Mute. Am I unmute now? Yes. Okay, thank you. Okay, can you hear me? Okay, yep. Thank you. Thank you very much for the invitation to present to your seminar series. So I'm going to discuss with you about our work on methods to build large-scale kinetic models. I think Joseph made the case why we need kinetic models of metabolism to predict cell responses, identify rate-limiting steps, and guide design of industrial organisms, or as Matthias presented, even to understand disease in the human age. So there are many kinetic models out there. Most of them, though, are of relatively small size, and despite the size, though, still there are some models that are very popular and has been used a lot in the literature and in research. So, but moving towards whole cell models will require the building of large-scale kinetic models. I think some of the issues Joseph touched upon as in the introduction. One large-scale kinetic model of high quality actually comes from Matthias and his co-workers, but this required an extensive curation in building this model, and they've done an excellent job on this. Now, the question is, what are the issues now if we try to develop large-scale kinetic models now? How many assumptions one has to make about the size and then the rate laws? Of course, we have to respect biophysical principles, especially elemental balance, to make sure that although we do not represent something, how we keep this information in the model and maintain some ability to expand and shrink in a modular way. And I would like to point here, as those that have worked with flux balance models, they know that elemental and charge balance is very important for the correct functioning of the model. So I'm going to discuss with you some of the methods we developed for a systematic reduction for the genome scale metabolic models. In building such methods or algorithms in order to reduce the such size of models, we have to have some objectives in mind. First of all, I will always like to build large-scale models reduced relative to genome scale, but still focused on certain pathways of interest. And then again, we have to make sure that we include all the information without making any ad hoc assumption. And while we minimize the network, is there a way to describe the information that will not somehow explicitly represent in the model? And as I said before, it's very important that we respect biophysical principles and make sure that we retain the major carbon flow. So towards this, we have developed an algorithm for such systematic reduction, which we call RegEM. I'll just give you here a kind of a quick run through this. This is the general workflow. We start with the genome scale models. Then there are certain stages, pre-processing, we do the network expansion, up to the stage of validation. What we call validation for a reduced model is a model that maintains a maximum consistency with respect to the parent genome scale model. And we can discuss this later if you want. I'm not going to go into depth about this, but I'm just going to show you here some examples here for the case of E. coli. The pre-processing starts by choosing the subsystems of interest that we have to include in the core model. Okay, so for example, we want to have TCA, pentophosphate, glycolysis, and then some other subsystems of interest. And then we also have to define what are the boundary conditions, what are the small molecules and also substrates, nutrients that the model has to represent, and we have explicit representation of the reactions and the pathways involved on this. And then what we do with the network expansion, then we start identifying all the reactions that are not included in the formal definition of the pathways, TCA cycle and glycolysis, but they connect in a pairwise nature in subsystems. So I include initially the TCA cycle as is defined in the curation of the model, generoscular model, and glycolysis. But then I ask the question, how many reactions do I have to introduce to connect these subsystems without introducing any additional metabolite? And we do this in a pairwise manner. And in this way, now we make sure that we include all the reactions that are not necessarily part of the formal definition of the subsystems, and yet they connect these subsystems. Here is the visual expansion. So that's how it looks if you start by a formal definition that you find in the genome scale model of E. coli. These are only the reactions you have for pentosphosphate glycolysis, TCA, and then we have some boundary. Then by asking how many reactions I have to include, then you start finding this kind of reactions which are not included in the definition, but they are necessary in order to connect the two subsystems without introducing additional metabolites. So this is what we call ultimately the D1, degree 1 of connectivity between the different subsystems. And then we have the degree 2. The degree 2, D2, is essentially if we now ask the question, how many reactions and metabolites we have to introduce if we are going to connect each subsystem by including one more metabolite that is not part of the initial system, and yet there are two reactions away. So we find all the connections that are two reactions away between the subsystems. And this is also a kind of snapshot from the human. We've done this also for the reduction of the human. This is the starting subsystems you have there. And after you do the D1 connections, you come to this particular size. Now the question is that after we define now the core subnetwork, what are we going to do with the rest of the reactions that are not included in the network? Towards this, we develop this next algorithm, which is called LampGem. And what we do with this is that we keep the core explicit representation of the core, but then we find what are the lumped reactions that we have to introduce in our network that can connect all the core metabolites with the corresponding biomass building blocks or other metabolic tasks that we want to describe in the model. So we start with the core metabolites, and we have the targets, let's say the biomass building blocks or any other of metabolic task. And then these are all the other metabolites and reactions in the gem. And then we find all the networks that connect this. For example, this is in the case of histidine. What I have here in black are the reactions that are part of the core model. And what I have in color here are the minimal subnetwork that I need to have in order to have balanced production of histidine. After I have identified all these subnetworks, these are not part of my core model, then I can lump, I can sum everything, and this way I can derive the lumped reaction that will represent the synthesis of histidine without including any new metabolite in my system except the core metabolites and the biomass building blocks. And of course, we can find alternative systems and alternative subnetworks to produce every biomass building block, and for each alternative subnetwork, I might have even a different lumped reaction. And we can do the same also for the human. So we start, we identify all the subnetworks that will take us from the core subsystems to target building, biomass building blocks, and derive immediately these lumped reactions for the system. For example, for the case of the core E. coli model, we go down to 102 plus 100 types of reactions versus the 2,000, more than 2,500 reactions in the genome scale models. That's how the D1 network will look in E. coli. And of course, if we can expand further the network, and you can go degree 2, degree 3, and this way, essentially, we can, let's say, move back and forth with respect to the size and the scope of the reduced model that we can have in the system. The same also applies for the human. And this way now we have a way to represent my core model. And you can see here how in the core model by then I can have also reactions that my algorithm told me that I must include explicitly in the model, although initially are not part of the formal definition of the starting subsystems that I have there. So now we can build the new generation of core models, which are manageable, scalable, and reproducible across the labs. And by the LampGem representation, we make sure that there is a way somehow to capture also the information on the reactions that we leave outside from the representation, and having some consistency with analysis that we have proposed, we can make sure that these models from the stoichiometric point of view are consistent with the genome scale models. Another thing which is also very nice for this is that now having an algorithm and having a definition of parameters, you can see now that every lab using the same genome scale model and the same parameters, then they can produce exactly the same type of models, and we don't have any more this problem that every lab can have its own reduced type of model. These are the basic papers from this analysis. Now, of course, moving towards whole scale cell models will require, for large scale kinetic models, we'll have to consider the uncertainty as both Matthias and Joseph proposed. And the uncertainty, of course, comes from the size and the nonlinearity, but also it's structural and quantitative. Let's consider the simplest case pathway, a branch pathway. If we even assume the simplest kinetics, reversible Michaelis-Menten for just the 5 steps here, then we can see that we need a large number of experiments in order to estimate very few parameters. And a lot of them must be dynamic experiments, and I think Matthias kind of presented some idea on what type of analysis and how much effort it takes. Now the question is, can we start building some kinetic models around these networks that we have? Of course, the issues, as we know, there is partial measurements with respect to the fluxes and the metabolites, and of course the number of the experiments. And as we all know, this leads to a system which is underdetermined, which means we have more unknowns than knowns. And towards this, we propose that instead of building a unique model in this respect, we can address the issue of underdetermined models by generating all the models that are consistent with a set of data. How we do this? So we have to apply a kind of sampling. And sampling, of course, is not new. It has been used a lot in the flux balance analysis, where the problem is the same. It's also an underdetermined system. We have more unknown fluxes than parameters. And towards this, we have been developing workflow, which we call Oracle. And the idea there is a workflow to build populations of kinetic models based on the partial information. And this is what is consistent with this partial information. And the trade-off here from the model uncertainty to partial data is that we have a multiplicity populations of models. Let's see how we're going to run this Oracle workflow around the simplest model. So imagine this simplest system. Imagine we measure only 2 of the fluxes, and then by doing some fluxomy, we can estimate all the fluxes in the system. So what we know here, we know the stoichiometry, and we have an estimate about the fluxes. So what do we need next is to know some kinetics. So for example, for the V1 or for all the kinetics in the system, we have to know the maximal activity, we have to know the thermodynamic displacement, and we have some idea about the enzyme saturation. So how are we going to do about it? So we don't know about it. If we have information about the reaction thermodynamics, about the equilibrium constant of the reaction, and measurements of metabolomics, we can estimate this number. However, this is not always the case because we have partial information about the metabolites even for the reaction equilibrium. So what can we do? Can we do some sampling to estimate this particular displacement term? So if I have some estimate, either experimental or general estimate about the ranges of the concentrations of these 3 metabolites, by sampling, I will normally sample the whole box that corresponds to this metabolite concentration. It turns out that if we apply some network constraints into my system, I can start using these considerations that come from the thermodynamics that tell me essentially reactions that operate in a certain direction should have an overall delta G that is negative. So this allows me now to start kind of cutting down and limiting my solution space or my feasible thermodynamic space. And this is how if I apply, let's say, the thermodynamic principles of reaction thermodynamic consistency with respect to the concentrations, we see now that the solution space or the feasible space of concentrations is much smaller. And even if I have partial information about the thermodynamic concentration, about the concentration of a metabolite, I can even have a much narrower definition of the feasible space of the concentration. Now I can do sampling in this particular area, and then I can have an estimate in a population line about the possible thermodynamic displacements that this particular reaction step can have. So what's about the enzyme saturation? The enzyme saturation, the nice thing is, first of all, the enzyme saturation is well defined. In this case, it's between 0 and 1. Now, if we have some metabolomic information or kinetic data, then we can impose this into my system, and then I can do sampling about the saturation for the rest of the components that I'm missing there. Am I missing all of them? Then I can sample for all of them. And then by back calculating, I can estimate. I have my feasible thermodynamic concentration here. I sample for the saturation. I can back calculate my Km. Once I have sampled for the reaction for the displacement, thermodynamic displacement term, and for the saturation term, then I can back substitute and I can estimate my maximum activity of the enzyme. So this way, upon successive sampling that obey the flux constraint and the thermodynamic constraint and the partial information I have, either about the metabolites or the kinetics, then I can derive again feasible ranges about the Vmax. And in this case, we can start building and having now populations of kinetic models that are consistent with all this data that I have in my system. Of course, now out of this, we can get a population of nonlinear kinetic models. We can do analysis in this population of models and derive some statistics. We have a very good idea about the thermodynamic displacement of the reactions, and we can better characterize the ranges of enzyme saturation and metabolite concentration. So we have built now kinetic models of this type, populations of kinetic models for E. coli, for yeast, for Putida, and for the human. And it seems that the method is quite efficient for a very simple reason that it works on a back substitution. Of course, now what is the challenge? The challenge is how can we make statistical analysis of these population models to quantify and characterize the uncertainty. We're looking in how we can use data mining methods in order to map the parameters to the properties. Of course, all these models are parameterized around a quasi-steady state observation. Now, the next step is to analyze and prune these kinetic models with respect to their dynamic properties. And of course, we are looking a lot to see how much the effect of the model size impacts the conclusions that we get with respect to these models, because we're very interested to understand, even just by looking at this population, how does my conclusions about the properties of my system with respect, let's say, to dynamic responses or to the rate limiting steps change if I move from the D1 to D2 to D3, different scale of model. Does scale matter? Of course, but what properties are affected as I change this time? The good news is that having this systematic way to build this equivalent kinetic reduced stoichiometric models and the kinetic models, we can make at least sure that we minimize the effect of the artifact of other kinds of assumptions that we do in the system. And of course, towards the wholesale kinetic models, the next step, as again Joseph touched upon, is the EMI models. These are the models that they connect metabolism and expression, and it has been a lot of effort in this area for years, and at least for the last 10 years or even before. We have seen a couple of EMI models are coming out, and we have recently developed a formulation, a mathematical formulation for the EMI models that allow us now to formulate the EMI models which have a certain type of nonlinearity, to turn this nonlinearity into the piecewise nonlinearity, and then use an MILP formulation, which allow us now to have very efficient EMI models that are also involve the minimum amount of assumptions in order to improve their computational capabilities. Also, we maintain the nice structure that you see here. So we have now the classical FBA metabolic models, but now we have a very nice representation of stoichiometric balance and constraint capacity constraints for the model. And we can see how we can now analyze and compare the difference of the EMI model with and without the constraints of the expression, and also by including thermodynamics. And I have here also for EMI model for Saccharomyces for yeast, and you can see here very nicely, if you had used only the flux balance analysis model that doesn't involve this feedback autocatalytic loop that you introduce by just putting there the mass balances for the expression, the glucose would be linear growing with respect to the growth rate. But once you start introducing the expression constraints and the thermodynamic constraints, of course, you come to a kind of saturation case, and you are consistent without making any assumptions without making any significant assumptions with the experimental data. For this, I would like to thank the group. These are the main people that worked in developing the reduced models, the reduced algorithm models, and the kinetic models and the ME models. And this is the financial support. And I would like to thank you for the invitation again and for the attention.
Thank you. Thank you both to our presenters. They're both extremely interesting presentations. Dr. König, was interesting to hear, on the progress of liver modeling. I think it was especially cool to see how you can track simulation results against experimental data. And Dr. Hatzimanikatis, it's fascinating to learn about how you're creating, this family of models to account for the uncertainty. Okay, so I'm going to open the floor to questions, but I think first we have a bit of a backlog of questions here. Okay, so first, Joseph Hellerstein had a question on Dr. Koenig's presentation. Joseph Hellerstein, you want to take it?
Yeah, just very briefly, with the complexity of the models that you're putting together, I was curious about what level of model verification is going on. I mean, there are things like, looking at mass balance, charge balance, block reactions, and I was curious about if they're also tested about, some of the dynamics as well. Maybe you can expand on that a bit.
Yes, definitely we have like the basic unit checking, mass balance, charge balance, and so on. It's all in, but also we checking against certain ranges where we expect the concentrations to be. So it's more like we doing our simulations, and then we checking if we are getting outside of certain cutoffs. And this is especially helpful for in the case of model fitting and so on, because basically we have then 20, 30 of these time courses under different conditions we want to fit. In the end, we get basically a list of tests which failed, which are just telling us, okay, our with our optimal parameters, the predictions are outside of the range which we tolerate as good. So we mainly use it for that, like basically to build like some constraints in form of tests on what we expect, how good our model should be. And this test, I would say, like this changed basically how we build models, because in the beginning we write many of our expectations in the tests, and a lot of things are failing in the beginning, and then we know exactly what we have to improve because we see the failing tests, and then we're trying just step by step to get better here. Yeah, but of course often it's difficult. Like a lot of things is like mass balance. It's not only on the individual reactions, but of course it's also things like if you put like 10 milligrams of a certain substance in the whole body, then like the sum over all tissues and all products and so on. If you do the molar thing, like all these sums over the whole body have to be constant, and over time you have to find 100% in the urine and the feces, and things like that is also tested.
All right, next, Dominic Steinitz had some questions. I can ask them for you if you'd like.
Yeah, I can ask them as well if you can hear me. Well, I was mainly interested in like you had acetaminophen there, or paracetamol as it's called in the UK, but which is very toxic and will kill liver cells. So if you're not modelling the liver cell population, isn't that going to? I mean, I haven't done very much work on this, but we did model, when I did do some work on this, we did model the liver cell population and its regeneration. So
exactly, this is like this idea. Like the interesting thing is that we can basically we can build hierarchical models and like hierarchies of hierarchies. So for instance, what we didn't do this for the acetaminophen and paracetamol, but like basically for the galactose, basically we can then replace our one tissue, one compartment liver model with basically sinusoids, which have like just the lining of the cells and have the zonation within the liver, and can combine multiple in parallel to basically then get the tissue basically getting this tissue zonation, because the necrosis or the damage of too high acetaminophen is basically in the central part of this sinusoids, and we basically we can model this easily by replacing a one compartment model with a tissue model. And this is also the idea. That's one of the ideas, like with this whole paracetamol data set that we go and then test in this multiscale models, the effect of too high doses, but also like of rescuing substances. It's basically like a glutathione adduct, which is very toxic. And if you like basically the missing glutathione is the main problem. If you give precursors of glutathione, like cysteine and so on, N-acetylcysteine, then you could rescue like the glutathione levels and remove this necrotic damage. But yeah, the idea is also going there, but we built already this template infrastructure, which I showed for the bone tissue models. We did it also for the detailed cell populations, but we haven't applied it to the paracetamol case yet.
Okay, thank you.
Thank you, and I'd like to open it up to anyone who has any questions. Well, I have a question for Dr. Hatzimanikatis. I'm curious to hear more about how, I guess, how you analyze a family of models. You mentioned that, I think touched on that at the end, but it seemed like you would expect them to behave very differently. So how do you, I guess, have like a predictive family of models, or what's used to analyze them?
Yes, so I'll tell you for a couple of the studies of systems we work. So there are certain types of data that you cannot include explicitly in the model. For example, some dynamic properties of the model. So what we do is that we build all these nonlinear kinetic models around a reference quasi-steady state. And then we perform the same dynamic response simulation as those that we have experimental data for. And there also we have partial information only for some of the metabolites. And then the first thing is that we prune the models. We keep only the models that are essentially are consistent with this additional information. So that's one of the things to improve, let's say, the predictability, because in this context, predictability means to reduce the uncertainty. Okay, so that's one of the ways that we have used to prune these models. Also, the other thing which is very interesting is that these models, although they're consistent with the reference steady state flux, the distribution of the timescales are quite different. Some of them, if we make, let's say, a stepwise perturbation, the time to settle takes more than a couple of doubling times. This means that essentially these models involve some slow dynamics, which in principle we wouldn't like to keep as a representative of the metabolism, which we know is in the time scale of the milliseconds. So this way essentially is a kind of further analysis that we can do in the dynamic domain that we can prove the models. Now, the other thing we have done in the past is that once you have the models and you provide some guidance to make the experiment, let's say if you want to improve the uptake of a certain substrate, then you attack these targets. Some of the suggested solutions are more successful than the others. And again, we use this additional experimental information in a cyclic way in order to further prune the model. And it turns out that we get even better convergence in the predictability of the population, but still are within the type of the population. And we treat this as a population average statistics of the responses of the model. Now, the other analysis that we do some parameter classification. This is another type of analysis, and we find out is there any specific property in the parameters of the models that are more consistent. And then, as you would expect, we identify which parameters in the model should require to be more tightly estimated in order to have a better prediction, rather than some of them that will be more sloppy parameters. So this way, again, you provide a lot of understanding and learning about a specific model, like in glycolysis and E. coli under these conditions, which are the sloppy parameters and which are essentially the more critical parameters. So in the next round, if somebody would like to start by estimating some specific kinetic parameters of certain enzymes under specific experimental, that would be the first targets that this model would suggest to start getting a better estimation in the parameters in the metabolites. So essentially, we try to ask this model, what information do I have to to have or to collect in order to reduce my uncertainty, and this will improve in the predictability of the models.
Oh, have you you raised, I guess, I think a fascinating point about like the oscillating cycle between modeler and guiding the experimenter and so on. Have you, I guess, have you been able to do this in practice, like to get the experimental collaborator in your own lab?
Yes, yes, we have done this, and we're doing this in two systems. One was in the Putida, the other was in yeast, and now we're trying to do this in the human metabolism. And again, we're very careful here to talk about prediction, because as we all know in the systems biology, this level of systems, complex systems, there is no prediction to the second decimal. So we should try to see how our populations can somehow capture of models, and the parameters can capture the most significant responses of the system, and that will be the ones that we will have higher confidence. Of course, as you increase the information, your uncertainty is decreasing and your confidence will be decreasing. So, one of the things, of course, as I've learned for so many years working with modeling and experimental data, both in industry and in academia, already a good model is the model that tells you what not to do. And I think many of the models of a certain quality, even not the perfect quality, can be very successful to tell you what is not the rate limiting step and what not to go after. So I think this is already a good prediction that we don't usually acknowledge. Okay.
I have a brief question follow-up along this thread, and that is, to what extent do you explore the reduction in the complexity of the model?
In what sense? Can you elaborate on this? I don't understand.
Okay, so the focus I've heard, and of course, with more parameters, ultimately you'll see, essentially the degrees of freedom in the data, and you'll be able to fit it great. So you could argue that, another part of the exercise of modeling is figuring out what parts of the model are actually not necessary, and to simplify the model itself. And maybe you can elaborate on your efforts along those lines.
Yes, thank you very much for this question. So this we approach this in two ways. Okay, one way is that we start with stoichiometrically even more reduced models. Okay, so essentially we take already when we do the stoichiometric reduction, the network reduction, we take much more reduced and much more expanded models. And of course, what is very key here to understand this lump gem algorithm, which is very important, you can apply it in any kind of ad hoc reduced model you can have, okay? As long as you define a core, the core of your choice, by having this lump gem, you lump in some complex, they look ugly, but they're very simple reactions that you can represent what is missing. So the next step that we're looking is how does the properties, of course, if I keep certain enzymes, certain explicit representation in my very reduced versus bigger model, how much perturbations in this particular step, in the parameters of this particular step, result to the same responses in my large and the small model. So essentially in this way, but what is very critical we found here is that you have to have a very systematic way in scaling up these models from the stoichiometric to the flux balancing, okay? This is the one part. The second part that we're looking is we'll go back to the very old and nice model reduction, dynamic model reduction, where essentially now we take all these nonlinear kinetic models and we apply methods from nonlinear reduction method of analysis, and this is work in progress, where essentially kind of I hinted before, we try to see, because a lot of this reduction in these models has to be time scale reduction, fast versus slow, and pulse, and there are a lot of very nice methodologies within the area of systems engineering that you can create, you can do this systematic dynamic reduction. That's what we're exploring now. So essentially going to these nonlinear models and asking the nonlinear model to tell me how I can do this nonlinear reduction. So one way is to build, let's say, go from the small to big, and the other is to go from big to small. I don't know if I answer your question, but these are the two. I don't know if you have any suggestions. We'll be very, very happy to share.
Yeah, I actually would be very curious to know a little more detail about these approaches from systems engineering that you've been using for model reduction.
Mm-hmm. Yeah. Okay, we can discuss this if you want this.
I also have a question. I found it very interesting building this kinetic models from the stoichiometric or from the FBA models. One common info, normally the information which is used for the FBA models is like on the one hand like the growth function, and on the other hand the gene deletions, single gene deletions, double gene deletions. If you have basically essentiality of this gene deletions, is this information used also for this kinetic model building, or could you use it? Because I could imagine these are like somehow additional constraints on your kinetic parameters if you can grow or cannot grow.
Yes, so as I said, when we do this reduction in the models, we have this validation step, or what we call consistency. So even before this, when we build the reduced models of certain size, we ask the question, how many of the reactions that I explicitly represent in my model, in my core model, that are essential in the genome scale, remain essential in the reduced model? Actually, they do remain usually because you have more constraints. The question is the opposite. How many of those that were not essential before are becoming essential? Okay, so this is the thing. Now, if we want to preserve this essentiality, then we can relax these parameters in the reduction, and we can ask what other reactions we have to include in order to give the flexibility in my reduced model, in order to preserve this non-essentiality that I have in my system. So by going back to your question, before we even go to ask this in the kinetic, first we make sure that we preserve or we correct the essentiality between the genome scale and the reduced model, and then we proceed to analyze. Because if it's essential in the stoichiometric, then it will be essential in the kinetic and vice versa. Right? So that's I think this is the point where you should address the issue of the essentiality, and the rest can become, the rest from without having seen it, but I would expect you might have some hidden essentialities coming into the model by the interplay of thermodynamics and kinetics, which you have not explicitly included in this model, where essentially, due to the thermodynamics, some reaction becomes unidirectional, and immediately some other reaction becomes essential because you constrain the solution space only in one direction.
Okay, I have one question.
Hi, how are you?
What's that?
I say, hi, how are you?
Yes, yeah, fine, thank you, yes. That was a very nice presentation. You're doing some great work out there in Europe. My question is, we're doing some work on Pudida, and your model popped up on our search, and we want to try and get hold of the model. So I got hold of the .map files. Is there a way to extract the model from those? Do you know?
Yes, I have. I mean, the models that we have currently, because the way we generate, we give them as MCA models. But
Right.
I think your student has contacted one of my colleagues, if I'm not wrong.
Yeah, correct. Yeah.
And they will be in contact to help you with this. So.
All right, great.
Right now we have not yet. We're building now the pipeline to make available all the nonlinear models. The problem with the nonlinear models is that we have not yet. Don't feel very comfortable with the pipeline, because when you have a nonlinear model, you have to do additional analysis. You have to make basin of attraction analysis. You have to make global sensitivity analysis. So it needs much more thing in order to have a kind of robust application of nonlinear models. And that's why we're kind of in the process of making sure we have the whole pipeline for the.
All right, then we'll we can hold on. It's great piece of work you're doing, though. It's I think you've hit the nail on the head on many things, actually, especially using the model to figure out what data to collect next. I think that is that's spot on, I think. So yeah, that's excellent. Thanks.
Yes, thank you. Yes.
Hi, I also have a question.
Go ahead.
On the Lamkim to Vassily as well. And I was curious in particular, I think you mentioned the application to human systems. And in human systems, in particular, the biomass reaction wouldn't have the same role as it has in bacterial systems. So I was kind of wondering if you could comment on the challenges of these model reduction approaches in human systems. In particular, can you just focus your reduction on the biomass precursor lumping the same way you do it for bacterial systems, or are there any caveats?
Yes, so first of all, when we build the reduced models and we build all the subnetworks for biomass building blocks, they are not flux models. They are effectively biochemical models, which means which subnetwork do I have to include and how I have to lump it if I want to represent this biomass building block. So before we go to even put some values in the fluxes there, the question is how I can preserve this. So the second thing that we do is that we're also looking at the list of the metabolic tasks as they are defined in building these models. And for the human model, depends on the group, and they're up to the 250 something metabolic tasks. And what we're trying to do now is to start developing essentially this, let's say we have the core model and we have a metabolic task list to find out all the minimal subnetworks that we need for each metabolic task. And all the minimal, what is the minimal size, what is the size plus one, etc. And this way we can create effectively for each metabolic task a list of subnetworks and the corresponding lumps. And then if somebody wants to choose between them, it can choose between them. It can include all of them. But this way, essentially, some of the metabolic tasks, it will be other functionalities of the human cell that to make sure that the human reduced model, it still has the reactions, either as subnetworks, we can also include the subnetwork, correct? Or as lumped reaction to fulfill this particular metabolic task. And so that would be essentially more of a library kind of thing that you can put together based on what you want to represent. I don't know if I answered your question, but
that makes a lot of sense, yeah, because I was wondering like how to do it for, say, yeah, indeed, the task the human cell has to do, but you basically would then use the same approach, but just instead of the biomass, which is the appropriate task for a bacterial cell, you would focus on the known tasks for a human cell basically instead.
Yes. So if you have these subnetworks or corresponding lumped reactions, then you can choose if you are going to include and which one to include. But again, it will be reduced relative to the tens of thousands of reactions in metabolism.
And you would still do it in a kind of a cell type specific fashion, I guess, as in using known tasks per cell type, which are different for a liver cell compared to a brain cell or something?
Correct. So what we're doing is that we do two things. One thing is that we do the reduction without any expression specification. So essentially we apply the expression data to make it tissue specific. And we're now trying to see if it makes any difference or if it will be useful to make reduced models that take simultaneously into consideration the tissue specific expression data so that we can make them cell type tissue specific, if that's what you're asking, but that's what.
Yeah, it's a kind of a possibility.
Yeah.
Okay, cool. Thanks very much.
Yeah, well,
I think it's time to end the presentation here. Again, thank you so much to both of our presenters, Dr. Hatzimanikatis and Dr. König. Thanks for taking time out of your busy day to come present. We really appreciate it. Thank you to everyone for coming. We hope to see you all next month where we have a seminar on whole cell modeling. Till then, have a good month.
Thank you very much. Thank you for the opportunity. Thank you everybody for joining. Yeah, and please feel free to ask any questions offline if you want. So thank you.
Yeah, thanks very much to both of you. Excellent talks. Thank you.
Bye.